This is Deborah Zaritsky with Grand Canyon University, Bio 515 with Dr. Ott, reporting on neural communication. The picture on my first slide here depicts what my assumption was of a neuron. I had thought that all neurons functioned by neurotransmitters such as dopamine, serotonin, and they bound to a receptor. That receptor then triggered a sort of response. I had known that such organs as our heart and brain and our muscles, particularly skeletal muscles, functioned on a sodium and potassium pump, but did not realize that that was not elicited by a neurotransmitter. So we have some just natural depolarization and repolarization happening. But before all that, we have just a resting potential. We know that when a membrane becomes less polarized, the inside becomes less negative than the resting potential at a negative 70 millivolts. We know that when our muscles contract and relax, that there is an exchange of ions, particularly sodium and potassium, which occurs, the process occurs within milliseconds. So we have that depolarization when we have an influx of sodium ions from our extracellular fluid come into the cell. And then we kind of become more positive there, and a hyperpolarization when our potassium moves out of the cell and we become more negative inside. We know that our voltage-gated channels are changing in response to our membrane potential. So along with our membrane potential, resting potential, we have two other ones. One is called a graded potential. So this is going to happen when we do have that exchange of our sodium and potassium, but we do need a trigger for this. This is going to be in a very specialized regions, and where our sodium channels are going to influx with our sodium. And we know this as excitatory. So when our muscles do contract, we're getting a triggered event, and then we have some action. This is a constant signal. We know that the stronger the triggering event, the stronger that active area is going to be with an impulse. The nodes within our heart, sinoatrial nodes, are giving out a pacemaker, kind of an electricity or impulse. So we are finding these types of potentials within our heart tissue as well. So our action potentials are going to be very brief and rapid. We have one-way propagation, very long-distance signaling, and this is going to come with just natural depolarization internally. These are always excitatory responses. Because they are excitatory in nature, our neurons would virtually overload if we did not have a break in between that depolarization and repolarization and hyperpolarization. So our refractory period is going to give us that delay and ensure that our action potentials are able to be given within our active regions. So just the anatomy of a neuron, we have our dendrites, which are going to be our location of what's called a postsynaptic region. And that is going to connect with what's called a postsynaptic place within our other neuron. So when we're discussing our millivolts and how our depolarization and repolarization, that's going to happen in a place of the axon. So when we start with an exchange of our sodium and potassium, that's going to happen within our nucleus. It's going to go down to these little anatomical parts called the nodes of Ranvier. And that electrical impulse is then going to go ahead and travel all the way down our axon. As we were talking about our action potential, it can go far distances. These are a lot less common than our chemical synapses. Chemical synapses are, as I had mentioned initially, they're going to be responsible for their neurotransmitters. So those neurotransmitters are going to have a self-induced change in permeability. So from one neuron, we're going to go ahead and take that from the presynaptic synapse. We have what's called a synaptic cleft in between. And then that postsynaptic is going to be on our dendrite of our next cell. So this is always going to lead to an action potential within our neurotransmitters. So I found that the neurotransmitters are going to be going through the chemically gated channels, but those chemically gated channels are either going to elicit a response within the neuron or they're going to block a response within the neuron. And as I was thinking about this, I was thinking about perhaps like, our external triggers, such as, drugs, alcohol. In a discussion question, we were talking about the effects of lidocaine and how they inhibit the sodium influx or the depolarization, and therefore they're going to increase the chloride ion there, and that will not allow a transmission of the impulse, and that lidocaine is what's going to go ahead and stop the pain, and that's known as an inhibitory response. But then I was thinking about, external chemicals that we're putting into our extracellular fluid. So if we were to break down, like the energy drinks, they are going to elicit more of that sodium and potassium response and allow more impulses. And, we had said that you're going to need a refractory period for those impulses, but, when we have such chemicals, as that, foreign drugs and alcohol, then that would, keep the neuron on overload, and I can see how that would damage a lot of our neurons. So in conclusion, this is what I had found. So our electrical conductivity transmissions are going to utilize the potassium and the sodium. They're only found in certain neurons, primarily the heart and the brain. We get those voltage gradients through depolarization and repolarization, which are going to go ahead and create our graded and action potentials. And these impulses are going to be transmitted along the axon, kind of going from one node to Ranvier, as it connects to our smooth muscle tissue and central nervous system. When we're talking about our neurotransmission, and such as dopamine and whatnot, those are going to trigger the graded potentials from the extracellular fluid. they're going to create excitatory or inhibitory responses through our presynaptic and postsynaptic structures. And this excitatory synapse will permit the passage of sodium and potassium simultaneously, while the other increases permeability of the potassium and chloride. So thank you. Interesting stuff here. My diagrams came from schoolworkhelper.net and zoology, while the primary bulk of my information was found in our text. Thank you.